Hi, good morning everyone, and welcome to the Health Data Research UK bi-monthly institutional webinar. So for those of you who've not joined these webinars before, you're very welcome. So we hold these every other month, and they are open to all HDR UK investigators, researchers, postdocs, tech teams, students, fellows, managers, and so on. The whole HDR UK family is very welcome to join this. So the aims of these webinars are for the HDR UK research community and wider community to hear about each other's research. We always discuss a topic of interest, and we also have a slot for news and updates from HDR UK Central, including upcoming events. So the purpose of these webinars as well is to help build that one institute ethos. We're a federated institute with sites and activities going all over the 4 nations. So it's really great to have this opportunity for us to come together to share information and have interesting discussions. We would really like this to be an interactive webinar as ever, so please do feel really free to ask questions as we go along, and if you use a Q&A facility for that, that would be the best format. So my name is Melissa Lewis-Brown. I'm the Interim Chief Science Strategy Officer at HDR UK Central, and I'll be hosting this webinar. We are very lucky to be joined by Claudia Langenberg, who's MRC Programme Leader at the University of Cambridge, part of our HDR UK Cambridge site. And Claudia will be talking to us today on is a research talk on her recent paper that she led on cross-platform genetic discovery of small molecule products of metabolism and applications to clinical outcomes. And we're also joined by Amanda, our Executive Director for Communications, Engagement and Involvement. And Amanda will be presenting our upcoming organisational strategy for our new strategy year. So we go with the financial year, it starts on the 1st of April. So for the 21-22 strategy year. So we look forward to hearing more about that. And then finally, last but by no means least, we have Sinduja Manohar, and Sinduja will be giving us a roundup of latest HDR UK Central news and any upcoming activities. So without further ado, I will pass over to Claudia for her research talk. Claudia, thank you very much for joining us.
Thank you so much, Melissa, for the kind invitation to present. Great, so I hope you can all see this. Again, thank you very much. It's a pleasure to be here today. Sorry, I'm just moving on. There we go. So let me start by just very briefly explain the rationale to you with this kind of schematic flow of biology. So there's what I would call a plethora of distal studies, so linking omic traits such as proteins or metabolites to diseases or the phenome here. And there are important considerations that limit our ability to infer causal associations from these studies. And that's because metabolites are very highly co-regulated and therefore correlated. And they're also influenced by a very large number of factors, external factors, and other factors that may account for any of the observed associations that we see. And they're also, furthermore, not disease specific. So what I would like to do today, just briefly outline some of the opportunities that arise from integrating genomic data and genetic regulators of metabolism in this case, so studying metabolites, to really better understand causal pathways to disease. Now, many of you are data scientists, and so you will not be surprised of the credo in genomics that big is beautiful. And so the very simple idea that's behind the study is that we wanted to maximize power for our genetic analyses by mapping metabolites across different platforms and therefore have the largest number of samples for any given metabolite that's captured. And we started very focused with a platform that's called the Barocrates P180 kit. So that capture is a targeted assessment of metabolites. And in this case, we started with a Finland, a local Cambridgeshire study, and we had 174 metabolites that were commonly captured across different groups here of metabolites. And so the trick that we used, as I said, by mapping across is here you see this metabolite split by different platforms that also capture subsets of these. So here an untargeted platform by Metabolon captures 38, and then NMR metabolites which capture an additional 10. So that's really where this cross-platform nature comes from. And this leads to this kind of slightly unusual study design where we have, and here you see kind of just exemplary, it doesn't list all 174, so those with similar numbers are kind of collapsed together, but here you see all the metabolites, and here's the sample size, and here in these colors you see the origin of the data or the results that we integrated. And the first 4 studies here are kind of Cambridge studies, so Finland, EPIC, and Interval, and then the bottom 4 are those that where we had results, and investigators generously shared their summary statistics in the literature, and we were able to map across. And there you see here, kind of by design, those metabolites that are ubiquitously captured across all the platforms we included, such as amino acids. Here at the top, the sample size is over 85,000 individuals, whereas if you look at here at the bottom, the extreme, biogenic amines that are restricted to the Biocrates platform, so you end up with only 10,000 individuals of the Finland study. And I should say that the majority of these data, as you can see for the first 4 studies, the majority of those had not previously been published, so it was new data. And the sample size therefore varied, and hence the power for each of the metabolites varied as well. So that was the design, and you know, as I said, metabolites are very correlated. So around 100 principal components explain the majority of the variance, and hence in this genetic study we used a multiple test corrected threshold of 5 × 10⁻⁻10 to indicate genome-wide significance. So what did we find? And I think I can say it very shortly, is we found a lot. So here this is and in some ways it's a feature of the design. It was the largest GWAS for each of the included metabolites, and the vast majority of the signal that we identified had not previously been described. So we describe 144 genomic regions and 499 locus metabolite associations. And you see this here, the results are displayed in this, what we call a 3D Manhattan plot. So here you see the chromosomal position. These are the metabolites kind of color-coded by their groups. So here amino acids and so on, the legend is at the top. And here is the p-value. And as I said before, so the p-value obviously depends on the power, and hence I hope you can make out that many of the very high peaks and extremely significant associations are seen for those amino acids, for example, that have the largest power. So what I want to point out, I can't go into everything in too much depth, but you see examples for kind of different associations. So we have some where you see a specific genetic locus, as we call it, that's associated very specifically with a given metabolite in a given class. You also see examples where a locus is associated with several metabolites within a class or a pathway. And then you see some, it's quite obvious here if you look across. So for example, here, kind of very pleiotropic signals here on the chromosome FADs, chromosome 11 associated with metabolites across all groups, and the same is true for GCKR and quite a few other what we call pleiotropic loci, so ubiquitously associated with many metabolites across different groups, and that has implications for how specific we can draw inference about the effect of this genetic location or the genetic regulation of pathways and metabolites, as you can imagine. So degrees of specificity is important to keep in mind. And then here, just a few other features, just to quickly outline. Here you see the allele frequency of the variants we identified. And here you see the effect size, and it's kind of color-coded by the possible functional consequences of each variant. And what you can see is here, so this dotted line here indicates to the left, what we call rare variants. And here this line is the largest effect of a common variant on a complex trait, in this case, body mass index. So the bottom line is you have a large number of variants with large effects for the rare variants as well as for common variants. So here being massively larger than what we have seen for other complex traits. We were lucky enough to be able because there's a question of the kind of genotype or imputation quality, if you look at chip-based or array-based data, and in collaboration with our colleagues in Cambridge from the interval study, we were able to validate those variants that were also captured in the exome sequencing of the interval study. So that's really, that's looking very promising. There were rare and common variants with very large effect sizes. And if you want to use those to draw inference on disease, that's obviously beneficial to start out with such extreme effect sizes. Now, at the right bottom is the functional annotation or the distribution of this. And in red here are these, what we call MQTL, so the metabolite associated variants, and we compared them to disease associated variants from the GWAS catalog, for example, for binary traits, so diseases, continuous traits, and all SNPs that we included in our discovery. And you see the large enrichment for non-synonymous exonic variants here. And that's not something that's restricted to metabolites. That's also seen for other omic traits. But just to kind of illustrate that again. So finally, these are kind of characteristics of these MQTAs or variants that we identify, where you may ask yourself if you're interested in translation. So why does this matter? And I only have time for 2 examples. They are more described in the paper if you're interested. So one thing we had observed, and my research interest is focused on diabetes, type 2 diabetes and insulin resistance. One of the things we knew that several of the metabolites that we investigated had previously been associated with the risk of type 2 diabetes. That doesn't necessarily mean much because many of them have been associated with type 2 diabetes. As I've said, it's not necessarily specific with regard to the metabolite, and it's not necessarily specific with regard to the disease. So we really thought to investigate whether the genetic discovery could give us new insights into the underlying pathophysiology or causal associations. And this is what I've just described as really what this graph highlights. I don't want to go into too much detail, but in blue here, these dots you see, these are the kind of metabolite associated variants. And this is for type 2, their association with type 2 diabetes. And you see this, what we call enrichment of these variants compared to a random set of matched, so characteristically matched control variants here. So matched because, as I've told you, these are not your kind of normal, well, not normal, these are a special set of variants with characteristics, as I've explained in the slide before. So you can't just use any variant randomly drawn. You have to kind of match them to these specific characteristics. But even if you do that, and if you're rigorous, you see this enrichment. Don't want to drill into too much detail, but one of the things that we noticed was that one of the very significant variants was a common missense variant in a gene called GLP2R. It's the glucagon-like peptide 2 receptor, missense variant that was associated here with higher citrulline levels. So citrulline is a known biomarker of intestinal function or gut function, and GLP2R is expressed in the gut and is closely related to the glucagon receptor and also GLP1R, which is the target of insulin-stimulating GLP1R agonists, which are used in the treatment of type 2 diabetes. And GLP1 is an insulin-stimulating incretin. So here, this is the Manhattan plot for citrulline. And so as I've said before, so we know actually that GLP2 stimulates intestinal growth, and it is used there's a condition called short bowel syndrome. It's used to treat this. And here I've highlighted a trial that has investigated teduglutide, which is a GLP2 analog, so mimics the effects of GLP2. And in this trial or rigorous setting, they've clearly demonstrated the effect on citrulline levels. And that suggests that you can use citrulline as a biomarker of intestinal function or target engagement, or in other words, GLP2R signaling. So the identified missense variant that we identified is in high LD, or is correlated with a different variant that had previously been described for type 2 diabetes. And what I want to show you here, if you look at this plot, this is the position of the gene. And here are these different traits, one over the other. And so we're zooming into this genomic region with which we saw the association with plasma citrulline. And what I want to just demonstrate by this, color-coded, is the correlation of the variants associated with each of these traits with our lead variant identified for citrulline. And what this highlights is that this overlap, what we see for citrulline and the variant in LD associated with type 2 diabetes, is not coincidental, but is a shared genetic association across these traits. So for citrulline, for type 2 diabetes, body mass index, and also fasting GIP. And GIP together with GLP-1 is one of the 2 major incretins. So basically it's the same shared signal. And then that leads us to the question of, so is this something that's specific to GLP2R, or do we see this also for the other regions that we've identified to be associated with citrulline? And this is what you see here on the right. So each of these here nearest or genes associated with this locus that we identified for citrulline, and this is their effect on type 2 diabetes. And what you can clearly see is that if you ignore ABO, which is a known very pleiotropic locus, GLP2R really is the only one that stands out. So it's a locus-specific effect, and it's not something that, based on this evidence, where I would say it's a circulating level effect of citrulline per se. So it's locus-specific effect. And the way that we interpret this is that the higher citrulline levels of the mutation or missense variant carriers indicate higher genetic GLP2R signaling, which in turn leads to chronically elevated GIP. So as I've told you, this is one of the two major incretins that stimulate insulin secretion. So it elevates GIP levels chronically, and in turn, over time, this leads to GIP receptor signaling downregulation in pancreatic beta cells, which then in turn leads to reduced insulin secretion and an increased risk of type 2 diabetes. And just to conclude this example, so GLP2R is a G protein coupled receptor, and this can this can signal either via G protein dependent cyclic AMP or G protein independent beta arrestin signaling. And this is something that we were able to investigate with our collaborators within the Institute of Metabolic Science, where our unit is located. This is Fiona Gribble and Frank Ryman at IMS, and they were able to express the variant in different in vitro models. And what you can see here basically is that the missense variant was associated with reduced recruitment of beta arrestin upon GLP2 stimulation. So this is this difference here. So at different concentrations of stimulation, you see here this reduced recruitment of beta arrestin, and you do not see an effect for cyclic AMP. So this in turn suggests that impaired beta arrestin recruitment to GLP2R contributes or may contribute or underlie the observed association with the increased risk of type 2 diabetes. So I'm going to conclude this example and just give you one final example, which is slightly different. It's also coincidental. So we, at the time of writing the manuscript, found a paper appeared in the literature, which talked about this disease here. It's a rare degenerative retinal disease, so an eye disease that's severe. Not many people are affected. As I said, it's rare. It's macular telangiectasia type 2. And this paper was a small GWAS of just a few hundred patients, and found, because it wasn't also particularly large or powered, now there's a larger study underway, but found a few, I think only three genome-wide significant loci associated with glycine and serum metabolism, or this is at least what the authors indicated. But at the time, it was not known whether this is coincidence or whether this is something that's specific, would be specific to other genetic loci that would be identified for this disease. Then more recently, actually, another paper came out, again, based on rare disease patients with this disease, that suggested that this low serum bioavailability leads to MECTEL through increased levels of another metabolite which isn't found in healthy individuals, but atypical deoxysphingolipids, and that this explains this. So this altogether kind of suggested that is serine metabolism, or that serine or glycine levels would have a causal role to play, but whether this will be seen more generally or whether that it could even help to predict people at risk of disease wasn't known. So we took our GWAS and all of the loci that we identified to be associated with serine levels, and this is and tested their association with MECTEL in collaboration with these people. It's a team led by Melanie Barlow in Australia, and this is what you see here on the right. So here you have the risk of the disease of MECTEL, or the association, in proportion, per genetically predicted difference in metabolite levels. And so each of these dots represents a metabolite, and we took all the variants we had identified for that metabolite, condensed them together, and then took that to look together to look at the association with the risk of MECTEL. And what you see is that this is the level of kind of multiple testing, and you see that really only two stand out at this very low level of significance, or high level of significance and low p-value, and this is serine and this is glycine. So what this suggests is that if you test these 174 metabolites, that this is very specific to this particular pathway of serine metabolism. And then you can now look whether this is proportionate to the effect of the variants. Now you're zooming into these two dots and ignore all others and all the variants that we identified. So is the effect on the risk of McTell proportionate to the strength of the effect of these variants that they have on the metabolite level? And that's what you see here at the bottom. And this is a beautiful dose-response relationship, all the variants associated with serine or glycine, and their effect on McTell. And what you can also see already here, this is the log odds ratio, is it's a massive effect. And so if you have such a strong effect, you can also ask yourself, does this in any way, or may this in any way help to do a genetic prediction of the disease? And that again is the figure here on the right. So this is the ROC curve, and in red you see the line when you don't include our genome-wide significant low surface serine and glycine, and then you see the added value or incremental prediction that you get over and above this baseline model. And there's also, I think, included age and sex, and you increase the AUC from 0.67 to 0.74, and that's a massive increase if you consider in some ways the ease and low cost of obtaining this genetic information. So in summary, I'd say this second example shows very strong evidence that genetically higher serine levels reduce the likelihood of McTell and can predict the development. And in fact, this is now Melanie Barlow and team and the McTell Consortium, obviously, who are experts in the disease, which we are not, but they've not only now done a larger GWAS, and we've done also collaborative follow-up studies with kind of cement or validate these earlier findings, but also considering the potential treatment effects, because if you can supplement serine in individuals at genetically high risks, this may be a promising treatment avenue and a low-cost treatment strategy in individuals at high risk, which obviously has to be evaluated in trials, but is very promising early evidence. So I'm going to end with this, and I hope I've shown you that this increased scale and greater variant diversity significantly increases the number of identified variants for all these metabolites, specific characteristics. We've shared all of our results in the web server that we've designed with our collaborators at the Helmholtz Center, and given you some of the examples we outline in the paper on the potential clinical utility and etiological insights. So if you're interested, please have a look at our website. And I'd like to just highlight, I have the privilege of presenting this, but it's been a huge international effort. So I want to particularly highlight Luca Lotter's and Mike Pietzner's work, who've really driven this to completion. Many other people, but particularly here, our kind of friends and collaborators from Munich, as well as a large group from Cambridge. This is Melanie from Australia, from the US. So thank you for the opportunity to present this on their behalf, and to all of you for your attention. Thank you.
Thank you, Claudia. That was fantastic, and a wonderful example of what can be achieved through national and international collaboration that could certainly not be achieved by any one single institute. Fantastic. Thank you for your leadership in this, not only in masterminding it, but bringing that incredible research team together. Okay, so over to Amanda White now. So Amanda is going to tell us about the HDR UK 21-22 organisational strategy, which begins, well, she'll tell you when it begins, 1st of April.
Thank you, Melissa. Can I just check on timings? Do you want me to shorten the talk?
Well, 10 minutes, leaving 5 minutes for Q&A would be fantastic. Okay,
no problem. Okay, so hello everyone. I'm Amanda. I'm the Director of Communications and Engagement at HDR UK. It's lovely to see you all on the call. I'm going to share my screen now so that you can see my slides. Okay, so this is our strategic delivery plan for the coming year, which starts on the 1st of April and goes through to the end of March 2022. The reason for sharing this with you today is, as members of our HDR UK community it's really important that you have awareness of our key priorities for the coming year, and where we'll be focusing our efforts, so that you can then align your work with what needs to be achieved strategically. But also, as members of our community, we really see you all as spokespeople for the organisation. So I know that many of you do lots of different talks and meetings and presentations. So it would be very helpful if you could use the key messages that we have in our strategic delivery plan, and we're very happy to share the link to this slide deck with you after the talk. So just a reminder and a recap, I'm sure that most people are familiar with this, but our mission is to unite the UK's health data to enable discoveries that improve people's lives. So we continue to have that big focus on how does our work translate to patients and the population. And we have a 20-year vision for any interaction, clinical trial, biomedical discovery, and public health to be enhanced by large-scale data and advanced analytics. So just to give you a quick recap of the process that we run through for our annual strategy cycle, this runs throughout the whole year, and the point at which we are at the moment is at the bottom of this slide. So we're about to publish, or we have published the strategy on the website, and we're currently in the process of sharing that across our community and with our partners. But we go through a whole process that includes listening to our partners, our colleagues, other stakeholders, to really inform where we should be focusing our priorities in the coming year. So to give you a quick summary of some of the feedback that we had, there's only one that's a direct quote on here, but the others are kind of aspects that we've summarised from the feedback. We heard through our research at the end of last year, which involved interviews and surveys with colleagues and stakeholders, that our work in particular areas, particularly with our work on COVID and with the development of the Innovation Gateway, has been really recognised and highlighted by our colleagues as important aspects that are helping us to achieve our mission and supporting those improvements in people's lives through health data research. So lots of feedback that we can really draw on to inform the future planning. I think the other thing to comment on is that people really acknowledge the role that we have at HDR UK as a convener in terms of bringing together different organisations and people to work together and collaborate. And I think that's really been a lot of the outputs that we've seen over the last year, and the impact of the work across HDR UK is because of that collaboration and that ability to work on projects at scale and on data at scale. So we've reviewed the One Institute strategy and determined the areas that we'll focus on for the coming year in areas that we feel make HDR UK distinctive. So this slide summarises our key messages, and if you are doing presentations, I would advise using this slide as a tool to really highlight and summarise what we do and how we work. So we describe ourselves as the only national institute for health data that includes England, Wales, Scotland and Northern Ireland. We are established to work across a wide range of health data, and we know that over the next 5 years, these data sets and participants will grow rapidly. And we're also catalysing and convening across the whole health data community, not only just with the organisations that are part of HDR UK, but much more broadly than that, so that we can play a leading role in international health data science. And we have these 3 themes in our strategy: uniting health data, improving health data, and using health data. And I'm sure many of you are familiar with this slide that we've had now in place for around a year, that shows how those 3 themes fit together to achieve our mission, and explains that the Alliance and the Gateway work fits under our uniting theme. The tools and methods and our health data research hubs fit under the improving health data theme. and the research discoveries and the training and capability building under the Using Health Data theme. And all of that is wrapped around the work that we're doing to build public trust and demonstrate public benefits, the communities that we're working with, and our governance. So just moving on to where we'll be scaling up impact in the coming year, with a focus on scaling up to sustain and build on the progress we've achieved through COVID-19 to enable discoveries across more diseases and on a global scale. We want to scale up a federated approach to improving data utility that's demonstrated by the hubs, and to support researchers across academia, in industry, NHS and charities in this area. And we want to be developing and embedding the use of the Health Data Research Innovation Gateway to enable this more efficient and effective federated health data research at scale. I'm going to go through in a bit more detail the areas that will be prioritising in those aspects. Just before I go into that, I think there was a comment in the chat earlier, which hopefully we can come back to, around diversity, but this continues to be a key commitment for us at HDR UK for the coming year. Not only in terms of the diversity of people that we have working across the institute and people that we're collaborating with, but also the diversity of data that we enable access to from more diverse populations to be more valuable for the research that we're enabling. One of the things that we're really looking forward to this year is the Black Internship Programme, which will be kicking off in August this year. And I think we have around 50 applicants that have been successful in that programme, which is really exciting. We're looking forward to making that announcement shortly. So just to go into a bit more detail on the uniting, using and improving themes. So the boxes on the left are what are the areas that are set out in our One Institute strategy, and then the areas on the right are where we'll be focusing in the coming year. So I've already mentioned about the federation piece. So this is a key part of our infrastructure strategy that our new CTO, Charles, is leading on for us. To enable that analysis across different trusted research environments within our community. We want to really build on the gateway. We're already showing that it's a very useful tool with over 200 data access requests already, and over 10,000 searches on the gateway each month for data. But we want to make that much more of a useful service through the gateway, enabling the customers that are using it to get the data access requests approved quickly and efficiently. And we want to really build this into a vibrant ecosystem that our partners can use. I mentioned the breadth of data sets. We've got ambitious plans to really grow those on the that are available via the gateway in the coming year. Looking at the availability of industry data sets through our partnerships, and also, yes, making sure more appropriate representation of the population. I think I'm running out of time, so I'm going to speed up a bit. On improving health data, you can see that in 2021, we're looking at more evidenced improvements to data. And developing the data engineering capability to get the data onboarded, linked, and improve it, plus working on those standards that are really important. And we're excited to be reviewing the second milestone of the health data research hubs over the next month, so there'll be more news about that in April. On the using the data theme, you can see that we're looking for really high impact outputs and innovations that are coming out of the data research, and really being able to share those and communicate those in a way that's really engaging, not only for our community, but for the wider sectors across the UK that are interested in this work that we're doing. And we're continuing our focus on training, so lots of exciting plans to improve that. And the added piece is what we're doing to build the One Institute. So for the coming year, we're going to be looking at a more joined up brand strategy across our different portfolio that we've developed in the last 18 months or so. And a big focus on our quinquennial 2 vision and review, which we're going to need your help with in the coming months to develop that. All the while, we'll be working with our values, our torch values, transparency, optimism, respect, courage, and humility. These drive our behaviours that we work to at HDR UK and are really vital to how we do our work. So on the transparency side, ensuring that our work is visible and clear and engaging to anybody that might be interested in having a look at what we're doing. We're going to continue to build our community. We've seen lots of growth in the last year and we want to build on that even further. So this slide's a nice summary of how we've got coverage now across most of the UK. And a new organisation chart, sorry, it's very busy, very small text on here, but hopefully you can see this, shows how all the various components of our organisation, including the national priorities where many of you work, and the health data research hubs all fit within our organisation structure. And finally, our governance structure as well, so that you can see how the different groups and boards all come together and enable the support of this strategy. And just finally, before I finish, I just wanted to highlight this slide that we're putting together at the moment. It's a bit of a work in progress, but showcasing how all of our work comes together to create this UK ecosystem for data research, and some of the outputs that we're already seeing, which are very impressive and have only been achieved by the whole community working together to collaborate. So we're starting to use some of these examples to really demonstrate the impact of HDR UK. I think we're moving on from the kind of creation of processes and the infrastructure, and we now want to move forward so that we can really showcase these impact stories in a meaningful way. So I hope that's been useful. I'll stop sharing and see if anyone has any questions.
Fantastic, thank you, Amanda. Another exciting year ahead in the HDR UK world. So there is a question around the Goldacre review. So a question around what will HDR UK's involvement be with the Ben Goldacre review on the use of health data?
Thank you. Yes, thanks, Malcolm, for highlighting that. So the review is underway for those of you that aren't aware. The government announced at the beginning, last month, that they've commissioned Ben Goldacre to run a review on health data research to make it more efficient and ensure safe research takes place. So we are, in terms of HDR UK's response to that, we want to be really supportive and encourage our colleagues and partners to respond to that review and to provide as much input to Ben Goldacre and his team as possible. We have been asked to be involved in some interviews by Ben and the team so that we can contribute directly. And we have some slides and key messages that we can share with colleagues. We're going to be doing that through our research directors and hub directors, if you would find that useful, so that we can all be providing input in a consistent way.
Thanks, Amanda. I'm going to encourage listeners, if you have further questions, to pop them either into the chat or the Q&A, and Amanda can potentially take those offline. Thanks, Amanda. That was fantastic. And I'm now going to hand over to Sinduja to let us know a bit about what's been going on lately in HDR UK and any upcoming activities that the community should be aware of.
Thanks, Melissa, and hi everyone. So yes, I will be giving an update on some exciting areas of work, and please do put in any questions in the Q&A function, and I will do my very best to answer them at the end. So next slide, please, Holly. So I'm sure it will come as no surprise, but there have been a number of updates on our work with COVID-19. So why don't we start off with that? A key part of our work has been to continue to support the UK's response to COVID-19. And I'm sure you'll see across our website and our blogs and website that researchers across the HDR UK community have very much been working on areas such as improving our understanding of the virus, clinical trials for treatments such as with the recovery trial, which I'm sure many of you are familiar with. They've also been involved in symptom trackers, getting a better understanding of the impacts on vulnerable groups, and most recently delving deeper into vaccines and things like the safety and the efficacy of vaccines. So as you can see from this slide, from the numbers from our latest report to SAGE, there is still a high number of active research studies. So please do keep an eye out for our fortnightly COVID-19 news roundup, which we try and promote as far and wide as possible to really find out about all of the latest insights. Next slide, please. So hopefully you will have seen in our updates or online, or even from the last couple of bi-monthly webinars, if you're a regular attendee, that there are 6 national core studies to support and accelerate the UK's research response to COVID-19. And we at HDR UK are working in partnership with the Office for National Statistics to lead the data and connectivity study. So we work in sprints, and for the last couple of sprints, we've had a lot of key highlights, including making 62 priority data sets discoverable through the gateway. There have been 15 data access requests that cover 63 data assets through kind of like a streamlined data access process. And just last week, you may have seen, we launched a call for applications from teams who can run a feasibility study to really establish a data flow that can give researchers rapid access to hospital admissions data. We also funded 12 urgent research projects that will be using national data to answer key COVID-19 research questions. So as well as developing their patient and public involvement and engagement plans to ensure that the patient and public voice is incorporated. You can see on the slide, they've also submitted their data access requests, and we're starting to see the impact of their studies. So for example, one of the studies led by Professor Aziz Sheikh used data to explore the effect of the Pfizer and the Oxford AstraZeneca vaccines in the community on preventing severe illness resulting in kind of hospitalisation from COVID. That was in Scotland. So this is a big area of research that really adds to the evidence base, and I think we're all looking forward to seeing further results from this, but also the other funded projects. And as Amanda mentioned with the Goldacre review, within our role in HDR UK, we very much want to support the review by highlighting the progress made by HDR UK and our partners, really in enabling the kind of efficient and safe use of health data. So whilst it is by no means at all limited to the work of data and connectivity, as there are so many other examples, these are definitely areas which we may look to highlight further as well. Next slide, please. So you'll see that we've also carried out a range of patient and public involvement work through data and connectivity as well, such as our vaccine research question prioritisation, supporting the rapid funding for projects to develop and implement their plans, having lay members shape our lay summaries and descriptions of projects and processes and things like, what a trusted research environment is. We'll also be looking to do more work to get our key messages out to the public to help them gain a better understanding around the access and use of data in COVID-19. So I'm sure we'll be able to update you more in the next bi-monthly webinar. But what's been really important is that we describe and are transparent about not only the work itself, but the impact of the work to everyone in our HDR UK community and beyond. So that really is just one way our comms team have played a big role. You'll see on the slide here that I've highlighted a few interview videos ranging from viral genome sequencing to vaccines to ethnicity and COVID-19. Personally, I've definitely found them very interesting and learned a lot, so please do watch when you can. Next slide, please. So moving slightly on to the Innovation Gateway, you'll see from the title of my slide, we're edging closer and closer to a big milestone of 600 datasets on the Gateway, which will very much be a huge achievement when it comes. I couldn't possibly go through them all, but there are a couple I'd like to mention. One is the dataset from the REACT studies. And for those that aren't familiar, the REACT studies have been working to improve understanding of how the pandemic is progressing across England. But there are also a number of datasets that are now discoverable on the Gateway from the UK Biobank. And Breathe, the health data research hub, have partnered with GSK to enable access to data from the Salford lung study, which is exciting in itself, but it's also the first industry dataset to be listed on the Gateway too. And the last ones that I wanted to highlight was actually something we announced only yesterday, so still hot off the press. Understandably, vaccines are on everyone's mind, and more research is needed to look at things such as effectiveness and the impact on transmission and things like that. So we're really excited to share that the England COVID-19 immunisations dataset and the Welsh COVID-19 vaccination dataset are now both discoverable on the Gateway too. So the rapid development of the system to kind of support this access is just another great example of what can be achieved through the Data and Connectivity Programme. Next slide, please. So moving on slightly, I just wanted to spend a couple of minutes sharing some great updates from the Cbd Coverage Uk Consortium. So for those that don't know, the consortium is a group of, I think it's more than just over 130 members across 40 institutes who are working to understand the relationship between COVID-19 and cardiovascular disease, and is managed by the BHF Data Science Centre. So the first update I wanted to share is that the Stroke Association have announced funding for research that will examine whether having COVID-19 increases someone's risk of stroke by using health information from nearly all UK adults. So this research will really build on the work of the Data Science Centre, which is fantastic to see. Another very exciting update I wanted to share is that the work led by the consortium and NHS Digital has enabled for the very first time a new linked health data resource that covers 54.4 million people, and it's now available for researchers from across the UK to collaborate. And it's a resource that links health data from GP records, from hospital data, from lab test data, and it really is the first of its kind. So I'm sure we're all intrigued to see the impact it could bring. Next slide, please. So in the last few minutes of this roundup, I just wanted to share a few different areas of work that might be of interest to you. Firstly, we're creating a new Gateway development and improvement group. So on the Gateway, we're constantly developing it, and it's important for a wide range of people to help shape this. And that's where this group comes in. So if you are interested, you don't need any previous experience, so please do have a look on our website for more information and to sign up. The second thing I wanted to highlight was around continued professional development for health data scientists. So the team are working to start rolling out a wide range of short online courses to help health data scientists add to their skills and knowledge. So do keep an eye out for more information. But if you are keen to find out more, the team provide regular kind of bulletins, so you can sign up to that. And lastly, we've been doing some work to really develop and grow our patient and public network called HDR UK Voices, and that's a chance for people to inform and shape our work. So whether you'd like to join the network yourself, or whether you have any patient and public networks that you can share this with, that would be fantastic, as we really want to make sure our work is informed by a whole range of people and views. Next slide, please. So before we finish, I just want to share some important dates. First is our Innovation Gateway drop-in session next Thursday, which will be a takeover by the BHF Data Science Centre. Another event of interest could be the researcher roadshows being held by the NIHR Research Design Service and their partners, and will be all about using health and social care data sets and research, and is particularly aimed at early career researchers. Our next Institute bi-monthly webinar will be at the beginning of May, so do keep an eye out for that. And lastly, but no means least, please do save the date for what I'm sure will be the biggest event of the year, the One Institute Conference, which will be in June. So thank you for listening and for having me today. And although it was brief, I hope it gives you an overview of the latest activities. I don't know if there are any questions.
Thank you, Sinduja. That was a great overview as ever, comprehensive. So yes, if there are any questions, please do pop them into the chat or the Q&A now. But otherwise, Sinduja showed various email addresses on the last slide. So do feel free anytime to get in touch if you have any questions around the strategy going forward, or any of the activities that Sinduja has just mentioned. Our door is very much open. Please do get in touch if you're interested to know more. So thank you to all three of our speakers this morning. Thanks very much for your time and all the work that you're doing. And thank you to everyone for listening, and hope to see you at the next bi-monthly webinar, as Sinduja said, on the 4th of May, if not at various other events in between now and then. So thank you, everyone, and see you next time.